Is Dr. Zan Schulten. Zan uses molecular dynamics to study the translation machinery, but today I think her talk is going to be a bit different. It's going to be on modeling methanogenesis. Zan.
That will be part two.
Okay.
Because in able to get to the point where, where we can model archaea, we had to start on a system where lots of information was known. And so we started with bacteria and E. coli. It's one of those model systems. And everything we did there, we test things that we want to develop, we tested there and moved on to archaea. But before I start, I'd like to really thank not the organizers, because that would be self-serving, because I was one of them, but actually, and I know how little we did, and how much the staff did that are sitting outside that really helped get this done. And also, Gene Robinson was so generous with his help and support. So those are really the people you need to be thanking. So what I'm going to talk to you about is our journey to get up to the point where we could simulate a host cell. And I think most of you know or are somewhat familiar with the methods that we have for modeling in biological systems. So we have everything from molecular dynamics. There have been molecular dynamics simulations on the ribosomes, and also DNA. They're sort of in this regime. They've been in this campus, we've been able to simulate an entire virus, so we've gotten up to subcellular structures. But I want to look at this region of single cells and complexes and whole colonies of cells. So I'm in the regime of microns to millimeters. So we have to give up these other methods, like of molecular dynamics, Brownian dynamics, and I remind you, they have time steps associated with them, femtoseconds, picoseconds. But if I'm going to do the cell and do something reasonable about the processes in the cell, I have to go up to hours. So the method I will show you is really coarse grain. It's called, most of the dynamics is described by a reaction diffusion master equation. I'll explain quickly what that is. But in giving up some of the resolution, we're able to gain ability to do very quickly reactions. But we now have to talk about the probability of the state of the cell being in a particular state. Our time steps are microseconds, and we will get up to hours and several cell cycles in our simulations. You notice there's a little grid over the cell now, because we're looking and dividing the cell down into various subvolumes to be able to do this. So this master equation, this chemical master equation, people might know it from chemistry. So if you have a well-stirred system and you want to look at how the reactions change in this, you might be describing about the system this way. For the cell, this P stands for the probability of the cell to be in a particular state. X is a huge long list of things that are in the cell, proteins, RNA, sugars, you name it. And these are reactions that take things out of a particular state of the cell, and these are reactions that take you into that particular state of the cell. Now, if everything were really well stirred, this would be a perfect picture. You can solve them stochastically, because there's often reactions going on in the cell that just involve a few molecules. But unfortunately, the cell looks more like this. In fact, this is based on a tomogram that I get from my collaborators on E. coli. And you're seeing here the dark spots, the position of all the ribosomes in this system. And they give me also the cell geometry, the position of the nucleoid region. And now we can look at a gene being expressed. It expresses, in this case, a membrane protein, and it can actually bring in more sugars into the system. So in order to handle the heterogeneity and the variation in particle numbers you see there, you really have to augment everything you do with as much experimental information as possible. So that's coming from single molecule tomography, people doing, cryo-electron tomography, single molecule studies. And we started a collaboration with Jamie Williamson to be able to get us the kinetics of the assembly of the ribosomes. So to these reactions, we add diffusion and another summation over here, because now we're looking at reactions in each subvolume and diffusion between each one of them. And I don't think I have to tell any of you that the cell is really crowded. If you go and look and put in all the particles that are there, you almost don't see any open space until you take a slice through and turn it on its side. So it's about 50% packed with something. So we try to include that in the simulations as well. And what I'll tell you about very briefly in the first part of the talk is our 3 different simulations that we did, which involved us watching E. coli for hours. And I chose these 3 because each one of them has an experiment that could then validate our simulations. So there's a little clock ticking in the corner here. You see we're already up to about 50 minutes there. This is only possible, this is on simulating an example of stochastic gene expression, because we have ported all of our software to being GPU based, and that allows us to get up to the time and length scales that we want to get at. I'll also talk about ribosome biogenesis. That's perhaps to date one of our most challenging systems, but at least we were able to capture the translational network. And then finally, we moved over into looking at colonies, where now what we're trying to do is capture the reaction dynamics and diffusion nutriments in and around in the local environments of cells in this colony with what's going on inside of the cell. And that really gives to a cooperative or emergent metabolic behavior of this colony, where cells on the outside behave differently than the cells on the inside. So just a quick summary. We try to put in molecular crowding. Where do we get this? In part from tomograms, cryo-electron tomography, from single molecule studies. We can also use information from RNA-seq and proteomics data. If we're going to do reactions, we do it most of the time with reaction diffusion master equations. That involves having kinetic parameters. Where do you get those? I'll give you a few examples. That's, again, we have to work very closely with experimentalists. And when we go to even the entire cell, what we would like to do is be able to use time scale separations and treat some parts of the cellular networks in steady state and then update them in a time-dependent fashion to the other parts. We've had some success with that, and that's where we're in the midst of trying to do with the cetaphrons. And finally, I'll talk just a little bit about our cell colonies, because one of the nice things about coming to this meeting is I've met a few colleagues that can actually are also looking at colonies of methanogens interacting with bacteria. So I think this will be an exciting new direction for us to go. So an overview, we have to bring in as much experimental information as we can. We build up our cell. We send it over to NCSA's Blue Water or a workstation with lots of GPUs. We run our simulations using our software, have it come back and analyze it with our VMD software. This is all software that we give out through our NIH center up at Beckman. And just to give you an example of setting up the cell, here's a tomogram from Wolfgang Baumeister and Julio Ortiz. So you can see, it gives me the geometry, the positions of the ribosomes, where the nucleoid is. And what's happening now, thank goodness, is you're really getting validation of this information from other methodologies. This is a single molecule experiment from Sunny Shi, where he labeled the ribosomes. He gets the same distribution. This is for the same E. coli, same growth rate, so it's really quite amazing. So just to give you an idea of how we set up, say, a small system like that genetic switch, I think most of you know this. It's called the Lac genetic switch. You have, here's the Lac gene, there's a repressor sitting on it. It has 2 binding sites for 2 molecules or inducers. If they come into the cell, bind to it, it goes off, and now you can start making the messenger, and the messenger makes is translated at the ribosomes, and it makes this permease, which allows more of the inducers to come in. So it can really switch on. Simplest model for this, it involves about 25 reactions, I think 8 or 9 species in it, and all the rates you can get through either biochemistry, single molecule, fish experiments. But we did end up having to correct one, because it does turn out whenever you're working on the host cell, there seems to be a whole community that knows about proteins and a whole community that knows about RNA. but they don't know anything about the other one. So we had really bad advice about one of the rates there, and we had to, once we corrected it, it then would agree with the experiments that were going from Sunny Shi. So this is what Sunny saw. He put a label, a fluorescent label, onto the permeases, and at a certain concentration of the inducer on the outside, he saw that roughly 1/3 of his cells turned on. They looked like yellow coffee beans. And even the ones that are dark have some of these permeases there. That's sort of the basal background. And you start seeing these coffee beans formed in about 30 to 40 minutes. So we put each of those bacteria, got its own GPU. We did randomization of the sort of what's the composition of things that are in the cells, and then let it run. And what you'll see as we start approaching 40 minutes is that about 2 or 3 of these cells will really turn on, and you'll see that they have lots of permeases. The little blips that you're seeing here is the messenger being formed, and then the messenger, he goes to the ribosomes make the protein, and the proteins are at the membrane, and then they diffuse along the membrane. So we're almost done with the movie. So those 2 cells have really turned on. This one's in the process of turning on, and these are sort of in their basal state. We can also do this as a function of different sizes. This is a slow growing. It tends to be longer and thinner, and you're watching the same thing. And down at this movie, you can watch now the repressors, and they're colored blue or dark blue, depending if there's 1 or 2 sugars on them. And the types of things that you can get out of these simulations are predictions of the lifetime of the repressor sitting on the operator, and also the localization of the messengers. Those are all things that can be tested. All right, so we have this snapshot of the ribosomes. So when I would show this work, somebody said, well, you know, in our first treatment, we made more or less the ribosomes stay fixed, or you know, their diffusion was so much slower than the other particles. So somebody said, I've really got to let those ribosomes free. And so now that was a big order to be able to do that. And we'd like to know how we get this distribution. And this started was a good time to lean back or go back to some work that I had done with Carl when we had looked at the molecular signatures in the evolution of translational machinery. So you've seen this beautiful picture of the ribosome, and you know, and I think we all know that Carl took the 16S, compared them, and saw signatures, sequence signatures, that differentiated the various domains of life. Well, in this PNAS paper that we did in 2008, we redid that study using now 100,000 of new RNA sequences in the Green Genes database, plus we did it for the large subunit, plus we did it for the ribosomal proteins. And these are where some of the major signatures regions are. And of course, we were really happy with this work. We thought, oh, you know, if that's a signature of a bacteria, wouldn't that be a good site to have a drug, an antibiotic, you know, kill the bacteria? And so we wrote a proposal to the Gates Foundation. It got rejected. But then the suggestion that we made and the particular modifications on some of the drugs that we proposed was actually done about a year later. I like to think it wasn't done by one of the reviewers, but I just feel good that our suggestion was very good. Anyhow, we were following what these positions were in the ribosome, what were their functional role in the ribosome, in its assembly, and our latest paper was done with some single molecule colleagues to look at the assembly, the early assembly units. And then I started working with Jamie Williamson, who had rates for the binding of all the proteins, or he had experiments from which you could work out what the rates would be. And so if you do that, stop. This is not supposed to be an eye test, but you start with the naked 16S. These are all the intermediates that come in until you form the 30S. Now, I'm only showing you about 145 of the most populated intermediates. And in fact, the strength of the lines here are indicating the integrated flux. Where's the most flux coming through? What pathway are primarily used? So we took this in vitro model that he had developed at 40 degrees. We put it inside of the cell simulations, starting with transcription from the operons, from all 7 of them, and then we simulated the formation of the ribosomes. And we started off at sort of a steady state, and we turned off the intermediates immediately and watched them form. You never see very many of them. They're always in small numbers, but we could track where they were in the cell, so we had people doing single molecule studies trying to follow those, and we went roughly from about 3,000 to 6,000 ribosomes. This is for a very slow-growing E. coli. Now, again, how do you know you're right? Well, just across the street in physics, there was a colleague, Tom Kuhlman, and his postdoc were measuring the ribosomes coming off of all 7 of those operons, and they measured them for E. coli with various doubling times. So we're looking right here, and here's the STORM and the qPCR data for this, for one of the operons D, and you can see it's around 1,000, a little more. Well, we get 6,000, they get between 8,000 and 9,000. So, and here's their STORM data, that you can see where they're positioned from one of those operons. So what did we get wrong? I mean, well, one of the things that we didn't take into account in the first, in that biophysical paper, was the fact that oftentimes, well, not oftentimes, always, there's DNA replication. So there's a certain number of these cells that don't have one copy of, say, that D operon. It actually has 2. So you can see the DNA being copied here. So you have to account, if you want to get really good connection to the experimental data, you have to take DNA replication into account. Another thing that was missing, we didn't have the messengers in there from the metabolism. And here's just a picture showing you sort of the shapes are roughly proportional to the number of proteins that are in those pathways. So we just talked about this part at the top. We talked a little bit about DNA replication, but we need all the rest of it in there. And we do know how to treat metabolism, at least in E. coli, extremely well, because of work that has gone on before by Bernard Paulson's lab, where he has really mapped out a very good model for the metabolism in E. coli. And what we now tried to do was to take this picture and connect it to the diffusion and reactions, and particularly we're interested in what comes into the cell and what comes out of the cell, and how does that affect the metabolism of cells at various places within these colonies. So this is what we did. We striped a single cell on top of an agar, watched it grow for 30 to 40 hours till it got to be about 1 to 2 millimeters in size. And then we watched the diffusion of nutriments, oxygen, acetate, succinate, so on and so forth around it, and then use that information to update the uptake into this metabolic network. And one of the things that you get out of studying the steady state behavior, the fluxes through these networks, is an estimate of the biomass increase, the growth rate. We could use that, again, to go back and say, well, these cells are expanding, they fill into the neighboring lattice sites, and now our lattice sites are no longer, say, 32 nanometers, like when we were looking at the single cell inside of it. They're more like 10 microns, where you have many cells in it. And then we just keep updating this process. So our time steps are still back into milliseconds, but to get to 30 hours, we're doing each of these things millions of times. So we really have to use our computers wisely. Here's a comparison of the colony diameter and its height that's from the theory, just allowing different packing densities, and comparing it now to various time points, where we've also used different labeled enzymes in these pathways. Some of them are going to be later in the glucose pathway, and others are actually, we're trying to look mostly at the acetate utilization. And this gives you a sense of how things change. At early times, everything is oxygenated, and then as you move to longer and longer times, the colony and the cells in the center are seeing less and less oxygen. Here you start with glucose. Again, glucose is in the agar, and also a certain amount gets into the cell colonies, but in longer and longer times, you're getting less and less glucose at the top. So now just a quick summary of what we see in those experiments is that after about 30 to 40 hours, we get a complete stratification of these cells. And because we have put in a two-colored plasmid, one looking at PTSG and the other one looking at the pathways involved with acetate secretion, we can see very clearly that these guys are consuming acetate. And what we do is then we go over and use the core facilities at IGB, and they have a microscope with structured illumination add-on to it, and we can look through and do a slice at 20 microns above the agar surface, and we should see in that case only the reporting from our GFP, and no red should be there. And if we get close to the agar, I hope you can see it, the green is just now on those really outside cells, and what we're seeing here is the M cherry, and we're seeing signs of the glucose consumption. And if you look at us slice through the side, you can see this profile agrees completely with our experimental prediction. And I'd like to point out, this was done, the calculations were done before the experiments. And so we're very, very pleased with this, and I think it also shows how important it is to be able to work with a good metabolic model. Okay, so now, where are we with the methanogens? So we had a grant with almost the exact same title. This was actually, I think, Carl's last grant. It was with me and with Nathan Price. And I was telling Larry, every time you get something from them, from a funding agency, they want you to do something for them. So I was in Washington, D.C., visiting them, and they ushered me into the head of the biology division, and he had two pictures above his desk. One was Darwin, and one was Carl Woese. So I came back home, and I told Carl this. I go, Carl, really, your picture's right up there with Darwin. And he looked at me and he said, why is Darwin there? So I think you all can appreciate this joke. All right, so where are we? We're getting at cell geometry. We're beginning to measure abundances of things that are inside of the cell. It's much more difficult. We do have a kinetic model for methanogenesis, and we just now finished collecting all the RNA-seq data, also the small RNA-seq data. This was really greatly helped by one of my collaborators, Bill Metcalf, but also the JGI gave us a grant, and they provided us with 100 more samples. So this is really enabling us now to build a much better metabolic model. So first of all, in a paper that was done with one of my single molecule experimental colleagues here, Tae-Kip Ha, who's also was at the IGB, Bill Metcalf, we did experiments where we imaged did a DIC images of the cells. We could then collect the data, get an idea what the average cell is going to look like. To be able to do abundances, save some of the proteins in there, we had to introduce labels into them, and then Tae-Kip Ha had a simple proteomics count where he would use antibodies and say, sort of like, this is for this protein MCR, it's got a SNAP tag on it, and it binds to here, and then they can calibrate, and we could figure out that we had about 10,000 ribosomes and say maybe 200 to 300 of these MCR molecules. Very slow. You know, the data that we have typically on the E. coli, that's actually done from fluorescence studies, and they'll measure like 1,000 proteins, or in yeast we have 4,000 protein distributions. So there's a long way to go yet. Now with this metabolic, with the metabolic model just of methanogenesis, we have been able then to do kinetic models of that, and helped in large part by data that's in the literature, but is particularly from Ralph Tower, who was originally wanted to attend this meeting, but his wife became ill and he couldn't come. But we're so thankful to him for the many rates that he's collected. So just to show you what we can do with that metabolic model, the experimental points are from Bill's lab, and the lines are from the simulations. So we're doing a really good job at getting the growth, the usage of methanol, the usage of TMA, and we get approximately the right ratio of the methane to CO2 production. We also learned a lot what of those steps in those pathways are really sensitive. So whenever we come up with the kinetic model, we also do a sensitivity analysis to determine where there be dragons. So here's our metabolic map. It started off first in the lab of Paulson, then it was improved here with Bill Metcalf and Nathan Price, but you still needed to further improve it because we couldn't even let in everything that was in this Ralph Wolf medium that they grow these things in. So we had to add parts over here. We also added, so it's mostly we had to be able to put in pyrrolysine and deal with O-phosphoserine and also improve some of the pathways that are here. So we're now on, quote, version 11 of this, so it took many tries. And some of the reactions that we put in, I feel really badly. We did this with Carl back in 2005, and that was to show the fact that to charge cysteine onto tRNA cis, there is the canonical pathway using a cis-RS, but there's also one where you start with O-phosphoserine. We even discovered this pathway, right? And then it was later validated in a paper with Dieter Soll, which is also a very interesting story, but I won't tell it here. It took us 10 years to get this into the metabolic map. So here it is. You can go the SEP-RS direction to get it charged or the canonical direction. And what's interesting is from the differential, from the expression data we're getting from our RNA-seq data, we can actually see that in some substrates, this pathway is preferred over this one for a factor of about 2. So it was important to get this in. The other thing that we did is we knew we could never get any of the knockout studies right on growth on TMA and DMA and so on the methylamines because we didn't have the ability to have the proteins have pyrrolysine in them. So this was a study that was done in 2006. They knocked out the gene that codes for the tRNA that loads the pyrrolysine there, and if you knock it out, they die, right? And so we needed that again. So here's the pathway that had to be built in. And I wanted to point out, here's now a crystal structure of the methyltransferase that's involved in all of those pathways involving the methylamines. And that pyrrolysine sits right in the middle. Now, unfortunately, we only could do an estimate of how many proteins have pyrrolysine in them because it hasn't really been well annotated in the genomic data. It has a stop, I think it's hijacked a stop codon, but you can sort of look for where those stop codons are occurring, say in the middle of the sequence, and do an estimate. So that's based on that type of estimate. So I'm sure there's more than just methyltransferase. So now we put in all these corrections. We again have to check the models with experiments. Here was some early work that was done back in 2012. And now we added all these corrections. And now we can also treat TMA, DMA, and MMA. And these are experiments. It was from the colleague on the previous paper, on the previous slide. So everything is looking good for these, that we're at least doing a reasonable job of getting the growth correct. And now that we have the RNA-seq data, and particularly the half-life data, we're able to go yet another step further. So this, I think, is the most fascinating graph, is that you look at the half-life of the messenger. This is genome-wide now, and you look at it on methanol, the cells being grown on methanol, acetate, and TMA. So look what happens here with the acetate. It's really slow growing. You really slow down the degradation, right, of these messengers. And so how can we use that information? I'll show you in just a moment, but what's also important is this genomic data is just not for the metabolic network. We can also get it across all these COG classes. And you see it's not uniform, how they affect these various pathways. So one of the things we did is we looked at the logarithm of the differential expression, say on TMA and acetate, and we compared it to the differential fluxes through here when you do the simulations on TMA or acetate. And if something is upregulated on TMA, it's green, and if it's downregulated, it's blue. So we just did a Boolean cut. So there's some variation in these, but you can see that there's a large effect. But when you look at the fluxes through here, you're picking up some variation, but there's a lot of variation in the messenger information that's not reflected in the fluxes. So this also does mean that we can, and these places here and here, you can guess where they are. They're involved in the methanogenesis pathways. But we can go in, and if we now start using this information about just one or two slides more. If I start using this information about the RNA-seq data, we can use that as upper bounds to the fluxes through various parts of the network, and then we'll get a much better predictive model. And as I said, that's what we're really after. If you get these individual cellular components to a certain degree of accuracy, you can actually make quantitative predictions that can be validated with experiments. So this is where we want to be. We've done translation, I admit, just on bacteria. Having done it there, we're hoping we can also come up with a good model and even a more simplified model for it in the archaeas. We know how to build in the metabolic networks, and if we can't, since things change at a different time scale here than they do here, we'll try to use time scale separation, maybe do it iteratively. And with that, we should be able to handle E. coli. We're also looking at yeast at the moment, methanogens, and some of the colony simulations lend them very easily to look at cancer cells surrounded by healthy cells. And what we're after is being able to have our systems respond to the local microenvironment and look how the cell responds in terms of its signaling, regulation, and translation. And these are the types of things that you have to measure. And one, I shouldn't end without showing you how much time this all takes. So, especially for Larry, right? So this is simulation volume. So here would be where E. coli would be. Here's yeast. Here's a red blood cell. And you're seeing a bunch of curves because this just depends on the lattice size that we take, whether it's 16 or 8 or 32, but also how many GPUs you have available. So, and this axis here means how many days do I have to get on the computer to get, say, 1 hour in bacteria or 1 hour in an archaea. So it's looking not too bad, right, if you get enough GPUs. But we dramatically shifted the slopes of these curves by using a new technology of writing the code. It's called just-in-time code generation, a compilation, and we can get a tilt of like a gain of a factor of 3. And that was all due to my very able programmer, Mike Halleck. And here are the people who really do all the work. You know, one was inspired by Carl. He really convinced me that I should look first at translation. That really got me willing to go tackle this whole translational network. And then people that are also involved with actually looking at translation are Tae-Kip Ha and Tom Kuhlman. They're both people doing single molecule studies here at the U of I. These are some of my fantastic students, people working on the systems biology, ribosome biogenesis model. We also were very generously helped by Yuri Alon at the Weizmann Institute because he sent us some of his bacterial E. coli strains with some of those enzymes labeled, really help us through our business. And of course, this is Wolfgang, who provides us tomograms. Elizabeth is giving us tomograms on yeast. There's Joe Peterson, and Seth Torr is also involved in this. And this is also one of the experts up at our NIH Center on GPU computing. There's Mike, and as I said, we're helped by so many different funding agencies, because it's almost like you can put an organism, on those 3 domains of life, you can almost attach to it an NIH or NSF or DOE funding call. So we're very fortunate to have all of them in place. Plus, I cannot say enough about the IGB, that they have these fantastic course facilities that even a theoretician like myself can go down, we can get training on their microscope. Then I immediately learn I cannot do this. So I hired an experimental postdoc who did it then. So, but he could use those facilities, and I'm really indebted to them. Thank you.
Ribosome assembly, do you have a good intuition what sort of prevents it from being clogged by partially formed complexes? If it goes from multiple directions at once, you will not really,
Actually,
the throughput will be very low.
we talked about that in the paper, and I had a bottleneck listed in the middle that you look at all the flux lines sort of go through there. So we looked at that with molecular dynamics, and you can understand what goes on there. I don't remember the details to give it to you, but that's what's nice. If you do the kinetic model, and if you start looking at blocks or bottlenecks in those, you can go back and use molecular dynamics to understand where is that difficulty coming from.
Same.
Uh-huh.
Again, on the ribosome assembly work with Jamie, how do you work it in the very crowded cell with all these pieces that need to find each other? Do you bias that a little bit, or are they just?
Actually, that's always the problem. Can you take the in vitro kinetic models you've developed, because he did it at much different concentrations, and can you just plop them into a cell at a much different volume? And so his ribosome assembly in vitro took about 15 to 20 minutes. When you put it into the cell, you get that 30S being formed in about a minute. I think that's the number I'm remembering. So we were happy with that, because we fully expected to have to go back and readjust everything. But we left that because people had measured this. Also, we're very lucky, the diffusion coefficients for the subunits as well as the whole ribosome have been measured by single molecule experiments. We could just use those. So we were lucky. And many years ago when we first started on the E. coli, we did a study how crowding affects, say, the rebinding of the repressor to the DNA. And we could see you could get up to a 20% change in the rebinding rates if you go to molecular crowding of about 20%.
They don't have trouble finding each other eventually.
No, and in fact, as I said, they form. We even see the intermediates. There's some that are formed very near to their messenger. We're hoping that Johann Elf, who measured the diffusion coefficients for the subunits, he's going to try and see if he can use the same technique to look at those intermediates. So I can't guarantee you all aspects of it are right, but the large features, like how many ribosomes we produce, are fine. We're below what they actually observed, but as I said, it's because we didn't take the DNA replication into account.
Thank you very much.